Good morning, everyone. Let's get started. Okay, so on Tuesday, last time we started talking about the interactions between between different alleles within one gene, right? So what is the relationship between different alleles, either the dominant or recessive? So what is the relationship between them? Like if an individual is a heterozygous, so something like that. After that, so we started talking about the relationship or interaction between two different genes. So when we talk about the relationship between two different genes, so we start from the basic one. So if there's no interaction between two genes, then we're gonna see the classical ratio, the 9:3 to 3:1 ratio, right? If these two genes are independently assorted on different chromosomes and they do not interact in any form. However, if there is a interaction or relationship between these two genes, we're going to see some variant from the classical ratio of 9:3 to 3:1. So we have talked about the complementation relationship. If two genes are if two genes complement each other, or if two genes are redundant. So if they are doing the same thing, so what is the ratio? Okay, so now let's continue. So we briefly talked about the third type relationship between two genes here. So this one is named the recessive epistasis. So I gave you an example to show this. So Okay, so we have two genes here, right? So one is the W gene and the other one is M gene. As you can see, only if the first gene, the W gene, is wild type, then you're going to see difference between the wild type M gene and the mutant M gene, right? Because only if the first gene, W gene, is mutant, it is wild type, and then the pathway, this part, the first step, is okay. Then the first gene W can transform the colorless precursor into the pink intermediate product. Only if this is true, then the second gene can either work on this pink stuff if it is a wild type, or it cannot. So you're going to see difference between the wild type M gene and the mutant M gene, right? However, if the first gene is mutant, so the first step of the pathway is blocked, you will not see any like pink intermediate product in this pathway. So no matter what genotype the M gene will be, so it doesn't matter because the first gene is a mutant one. All right. So in this case, let's see what is the relationship between the classic 9:3 to 3:1 ratio to this one. Because of this recessive epistasis relationship between the W gene and the M gene, all right? So these two classes will be grouped because they show the same phenotype, the white flower, all right? Then we can group these two. This becomes 4, right? All right. So if you see this ratio, so immediately you need to know, okay, so this is a classical recessive epistasis example, right? Okay, So, okay, so this is the term, the new term for us, so epistasis, which means the standing upon. Okay, in this example, we have W gene and M gene, right? In this case, the W gene is actually standing upon the M gene, all right? And because this is a recessive epistasis, so only if the first gene, W gene, is a mutant, and in this case, the M gene will be hidden. So, okay, so because the first gene, the first mutant gene W will override the mutation for the second one. All right, so this is the recessive epistasis, right? Okay, this is another example. As you can see, the same ratio, right? The 9:3:4, all right? So in dogs, there are two genes. One is the B gene, the big B and the small b, the capital B and the lowercase b. And so this gene determines the color of the hair, the coats, all right? The big B will give a black coat for the dogs, and the little b will give a little bit like less dark, so brown coat in dogs. All right. However, so the phenotype of this gene is totally depending on the second gene. So there's another gene, which is the E gene, right? So the E gene, we have a capital E versus a lowercase e. All right, let's see what is the function for this E gene, right? Is it okay, so the E gene actually determines the deposition of the pigment in hair, all right? All right, so only if this gene takes the wild type, capital E, and then the pigment can be deposited in the hair, in the coats, right? If this gene is a mutant, so no pigment can be deposited in the hair. So this will show you the yellow color. So okay, so only if we want to see the B gene is expressed, so the B gene will be expressed only if the E gene is a wild type. Otherwise, no pigment will be deposited in the dark hair. Right. So as you can see here, okay, only if this gene is a wild type, okay, and this will determine, okay, only if the E gene is a wild type, and so this lowercase bb will give the brown color, and the big B will give the black color. Okay, however, if the E gene is a recessive, so this is the recessive epistasis, right? So if the E gene is a recessive, then this will give us a uniform color, which is the yellow, because no pigment can be deposited in the dark hair. Right? So this is another example to show the recessive epistasis. All right, yeah, please remember this classic ratio here. Okay, 9:3:4. Okay, now let's talk about the fourth type of the relationship between two genes. All right, so this type is called the dominant epistasis. All right, the third one is the recessive epistasis. This one is dominant epistasis. So what's the difference? Okay, all right, so in this one, okay, only the recessive, the first gene, the E gene, only if the first gene is a recessive, and this gene will override the second gene, right? Okay, now in this case, if the first gene is dominant, then the first gene will override the second gene. So that's why you're going to see a different ratio here. So this is the, in this case, the ratio will become 12 to 3 to 1. Okay. All right, so this is an example for the dominant epistasis. And the ratio should be 12 to 3 to 1, all right? So we have two genes here. So first gene is the D gene, and the second gene is W gene, all right? And for each gene, there are two alleles. The big D is greater than, is dominant to the lowercase d, and the capital W is dominant to the lowercase w. All right, let's see what the functions of these two genes are, all right? So the W gene actually, all right, so let's look at the D gene first. So the D gene allows the plant to, so actually the capital D gene will make the petal, the flower petal, darker. So that's the red pigment. Okay, so the capital D will produce the dark red pigment. So the little d, which is the recessive, the mutant, will give small amount of the pigment. So then you're gonna see the difference, right? This is the dark red and this is the light red. So this is determined by the capital D and this is determined by the little d, okay. However, this D gene can only be functioning only if the first gene, which is the W gene, is a recessive, okay? Which means like, okay, only if the W gene is a lowercase. So in this case, in these two cases, the W gene should be like this, the little lowercase w and lowercase w. If W gene is a wild type like this, or heterozygous, so it will prevent the expression of the D gene. So that's why you see if we do the dihybrid cross for this example, if we cross this one, with the same genotype here, the dihybrid cross, we're gonna, theoretically, we're gonna have like 9 to 3 to 3 to 1 ratio, right? Okay. However, only these two cases where the W gene is the lowercase w. So only in these two cases, the D gene can be expressed. We can see the difference between the little d and the big D. However, for this, you cannot see the expression of the D gene because the dominance allele of the W. Okay, so this will give us the same phenotype, white. All right, yeah, I hope this is clear. So in this case, the dominant, it is called the dominant epistasis. All right, and this gives us a little bit different ratio, 12 to 3 to 1. Okay. All right, so this slide just summarize the biology behind this example. The ratio is 12 to 3 to 1. And if we do the dihybrid cross, we're gonna get the 9 to 3 to 3 to 1, right? Because only in these two cases, oops, not this one. Only in these two cases, let me erase this, sorry. Only in these two cases, the W gene is a, oh, geez, sorry about this. only in these two cases, right? No. WO, my apology. Only in these two cases, the W genes are the recessive. So this will show the difference between the D genes, right? But in these two cases, we have the wild type W. So this will totally like hide the effect of the D genes. So these two groups will be clustered together. This is 12. So this will give us the ratio of the 12 to 3 to 1. All right. So any questions? Okay, good. All right, so actually, yeah, this is the biology behind this example. All right, so we start from, so in each plant, so there's a colorless precursor. Okay, we have two genes here, so the D gene and the W genes, right? All right, so, okay. And so this W genes will, so if there's a wild type W genes, this gene will restrict the pigment to only be deposited in these dots, not the petal. So if this gene is a wild type, so you only see the white color petal like this. So this is 12. However, if this gene is a recessive one, okay, now so the color, the pigment can be deposited in the petal like this. Then only in this case, we will see the difference between the big D and the small d, because big D will produce the dark red and the little d will produce the light red. So only if this W gene is a recessive, it will not restrict the pigment to be deposited in the petal. Then we're going to see the difference between these two alleles for the D gene. So that's why, so this is the biological pathway that explains the ratio of this 12 to 3 to 1. Okay. All right, the same thing here. So still, you can see the ratio of 12 to 3 to 1, right? So in squash, all right, we have two genes. So one gene is the W genes, right? Okay, so the wild type W gene will is actually is a inhibitor of an enzyme. So if this gene is a wild type, so it cannot transform the white precursor into the green intermediate product, all right? Then, okay, you only see, so if this is true, so if this W gene is a wild type, you only see the white color for the squash. However, if the W gene is a recessive, so which means like this pathway will not be inhibited. So this kind of genotype can transform the colorless precursor into the green stuff, the intermediate product. And then only in this case, another gene, Y gene, can be functioning. So this Y gene will either, so the capital Y will produce yellow, and the lowercase y will produce green. Okay, so this is another example to show the dominant epistasis of the ratio of 12 to 3 to 1. And yeah, this is the two genes pathway that influence, that determine the coloration of the squash. Okay, so we have learned 3 cases here. So it's very clear how we can get this ratio, right? Because of the dominant epistasis. All right, the same here. So when we look at the color of the feather of the chicken, so also yeah, shows this ratio. And this is another example. Okay, the 5th one is called the dominant suppression. dominant suppression. And this, yeah, for this type of relationship between two genes, we're gonna see a little bit different ratio, which is 13 to 3. So it's very easy, right, to understand this. Okay. All right, if we group this with this, yeah, this will give you 13. And this is the 3. Okay, so let's see what is the biology behind this one. All right, so first, suppressors. So what is the suppressors? Suppressors actually refer to a special class of mutation, okay? Those are the mutations that can reverse the effect of another mutant gene. Okay, we have talking about, so here we are talking about two genes, right? Let's say we talk about A gene and the B gene, all right? Okay, so if the first gene is mutated, you're gonna see the mutant phenotype. However, all right, so if B gene is also mutated, okay, if the mutation of the B gene can reverse the mutant effect for the gene A, then we say B is a suppressor. Okay. Still, we are talking about two genes. Okay, if the mutation of the second gene can reverse the mutant effect of the first gene, then the second gene, the B gene, will be called the suppressors. Okay. So let's look at the example here. All right. Okay, so this is still a flower example. Okay, so we have a P gene and we have a suppress and the SU gene. SU gene means suppressor. Okay. Okay, let's look at the first gene, the P gene. The P plus means the wild type, and this allele will produce the purple pigment in the petal, flower petal. If this gene is mutated, so we're gonna say this is a P gene without plus, and this will give the white color flower petal. Okay, so we have two parents here, so we cross these two parents, and we generate the double heterozygous F1 progeny, right? And then if we do the intercross for the F1 generation, we're gonna produce F2 population, right? And classically, we're gonna get the 9:3 to 1, 3:1 ratio. Right? So let's look at each of these four categories, classes, separately. Okay, so for this one, the first one, because the P gene is a wild type, so we're gonna see the purple petal, right? Flower petal. Okay, that makes sense. All right. Okay, now if we see the second category, so this one is still a wild type. All right. If the second gene, the suppressor gene, the mutant will not have any influence on the P gene. If the P gene is a wild type, then we still see the purple flower, right? Okay, let's look at here. This one. So both P alleles are the mutant. However, if the suppressor is also mutant, the two alleles of the suppressor gene are mutant, then this mutation of the second gene, the suppressor gene, will be able to reverse the mutant effect for the P gene, all right? So in this case, we still see the wild type phenotype, which is the purple flower petal, due to the mutation in the suppressor gene. So we're gonna talk about this, why this is like this, all right? Okay, so since these three classes are showing the same phenotype, so we can group these three together. So this gives us 13, right? Only this type, okay, let me erase this, messy here. Okay, let's look at the last class. Okay. Okay, now this is a mutant, but this is wild type. And this wild type suppressor gene cannot reverse the mutant effect of the first mutant gene. So then this will give us a different phenotype, the white petal, the flower petal. Okay, so this gives us a beautiful ratio of the 13:3. So in this type of relationship, which is the dominant suppressor mutation, it can also, so it can take two different ratios. The first is the 13:3, and the second one is the 10:6. 10:6 is what? Yes. So if let's look at it here. If the suppressor genes are mutant, however, if the first gene is a wild type, then if the mutant suppressor cannot have any effect on the wild type P gene, then this will also give you the mutant phenotype. So if this is white, then what happens? Then this will give you a slightly different ratio. We're gonna have like 10:6 ratio. So I hope this is clear. Okay, so in the exam, if you see like the ratio like this, 13:3, or if you see the ratio of 10:6, then immediately you should be able to know, okay, this is because of the dominant suppressor mutation. Okay, so this is the biology behind. Okay, let's see how this dominant suppressor mutation works behind the working example, right? So suppose, okay, so this is the wild type. Okay, and the wild type, we have the first gene which produce a protein with a specific shape, like here, okay? And we have a suppressor gene, which is S. The wild type will produce a specific enzyme. And this enzyme has a special shape, and this shape can perfectly match the shape of the product from the M gene. And the integration of these two proteins and the complex of this protein will be active, become an active protein complex, and this can produce the normal phenotype. All right, this is the standard situation. All right, we have M gene, wild type, and we have the suppressor gene, wild type. And if both are wild type, then they will produce a complex, active protein complex, which will yield the normal phenotypes. Okay, now let's look at the first situation. If this gene is a mutant, all right, but the suppressor is still wild type. Okay, what happens here? So the mutant M gene will produce a slightly different protein. So we say because of the mutation, so the shape of the protein, the product has changed. See, it's different. So this cannot match the shape of the product from the wild type suppressor gene. So it cannot. So these two protein cannot form a active protein complex. It's inactive. Then you're gonna see what? The mutant phenotype. Does that make sense? Okay. The same thing here. All right. So if the gene is a wild type, but if the suppressor is a mutant, so okay, the same reasoning behind. So these two protein that do not match in terms of the shape, and this will be inactive, and you're gonna see the mutant phenotype, right? However, if the first gene is a mutant, it produces a product protein with a slightly changed shape. However, if the suppressor gene is also a mutant, and it can produce a slightly different protein with a changed shape, and they can match. Both mutant protein can match, and they can form a complex which will become a active protein complex, and this will gives us a normal phenotype. All right, so this is the biology behind, and this gives us like either the 13:3 ratio or 10:6 ratio. Okay. All right. Oh, yeah. So there's a question. So is it possible for both mutant M and mutant Ls to mutate, but not come together? Yeah, of course, that makes sense. But so we are talking about the specific situation. So if the mutation of the M gene and the mutation of the suppressor gene, if their product can match to form an active protein complex, then this will give us this ratio or this ratio, right? But if in the case where you just mentioned, so if both mutant cannot come together, cannot get along together, then yeah, it will not show this ratio. I hope I answered your question. Yeah. How can a P plus be white? Oh, yes. Okay. How can a P plus dash and the suppressor be white? Yes. So in this case, oops. Let me erase this first. How can this be white, right? All right. So first, if this is true, if this is white, then this will give us this ratio, right? So how can this be? Okay, of course. So if this P plus produce a protein, it cannot function by itself. It must be in a situation where there's a enzyme that combines to this product from this P plus to form an active protein complex, then it will produce the pigment, purple pigment, to produce the wild type phenotypes, right? Okay, now if this is a P plus, but the suppressor is mutant, so it cannot produce any like useful enzyme to bind to this protein, then this will give us the white phenotype. I hope this helps. Okay. Yes. So yeah, SU is a mutant. So if it is wild type, should be SU. Oops. SU plus. Okay, that's the wild type. Something like this. So here the S plus is equivalent to the SU plus. Okay. Okay, very good. Okay, the last type. So we have a total of six different relationship between two genes, right? The last one is if the two genes are interacting in different pathways. So this will give you the classical 9 to 3 to 3 to 1 ratio. Why? Because if they are interacting in different pathways, if these different pathways are not affecting the same phenotypes, then we can treat these two genes are independent, right? So this will usually gives us this classical ratio. So for example here, but sometimes they will. So this is easy. If these two genes are totally independent, it will give us different phenotypes categories, and which shows the 9 to 3 to 1 ratio. However, sometimes even if two genes are involved in two different pathways, However, they can show a slightly different ratio like this, 9:6:1 ratio Y. So this is example. So if these two genes are having a cumulative effect, for example, okay. See, oops. So, excuse me, would you please mute yourself? Because I can still hear some background noise. Okay, thank you. Okay, let me discuss this first and try to, let's see. Okay, good. So how can I? So Amy, do you mind? Okay, good, thank you. Amy, do you mind if you can mute yourself? Because I cannot get rid of this window from my screen. Thank you. Okay, let's put it here. I don't know why I cannot mute, because am I the, let me see. Okay, let's do this. Okay, so in this example, even these two genes, A gene and B genes, are functioning in different pathways. However, if they have a cumulative effect, so for example, both A gene and B gene will determine the length of the squash, So the wild type A will give like long shape of the squash, and the wild type B will also like accumulatively at the length of the squash. So if this is the true, you're gonna see the double elongated squash. Okay. Okay. Now, if only one gene is a wild type, either A or B, so this will gives us the spherical shape of the squash. Okay, now if both genes are mutant, then it will give us a disc shape of the squash. So this, in this case, you're gonna see this ratio, 9:6:1 ratio. Okay. Okay, so there are some other examples here, so I don't have time to go through this. So you can, if you are interested in that, so you can read the slides later and also read the textbook. So it's fun to read those. All right, so this slides actually give us a summary about the different ratios. All right, we start from like the classical 9:3:1 ratio, right? All right. So in this case, there's no interaction between these two genes. Now we're gonna see this. All right, those are the different, the variant of these ratios. And it's very important for each of us to understand these ratios and how these ratios are formed from different situations. So we have covered like different types of relationship between the two genes, right? I hope, yeah, this helps. Okay, so I think, yeah, this is also the summary here, okay. this is the final slide for this lecture, for the lecture 10. Okay, so even we talk a lot about the genetics behind either one gene or two genes, but do not forget the environmental factors also should be accounted in, right? Because the observed phenotype, any observed phenotype is actually the consequence from the joint effect from genetics and environmental factors. Okay, even for some genotypes, so we're supposed to see some phenotypes from this genotype, but it can change because this genotype, this association between the genotype and the phenotype also depend on the environment. So if you change the environment to grow this individual, probably the association between the genotype and the phenotype will be destroyed due to the different environment. Okay, now so I would like to re-emphasize these two concepts here. Okay, so the penetrance rate and the expressivity. So even for the same genotype, you could see different phenotypes. You could see unexpected phenotype because of the environments, right? So this is why we, for the same genotype, probably we're going to see the variable penetrance and also the variable expressivity, or both. Because these two concepts are important. So probably in the next midterm, I'm going to give you some questions to test if you understand these two concepts here. All right. Let me see, what is the question here? Is that the long, oh, okay. Maybe, let me go back to the slides. I don't know, maybe, yeah. There was a typo. Oh, sorry, totally opposite. Thank you for pointing out this. All right. Sorry about this. All right, this long shape will be the double mutant. All right. So okay, so either the A gene or B gene, if either A or B is a wild type, so this will give us the normal shape, the spherical shape. If both genes are wild type, and this will give a lot of pressure on this individual. So this is a disc shape, all right? If both genes are mutant, then yeah, the squash will be doubly like elongated. Yeah. Sorry about this. Thank you for pointing out that. Sorry. Okay. Very good. All right. So I think I'm done with this lecture. Let's continue the other one. The lecture 11. Let me stop sharing here and continue with this one. Okay. Okay. All right, now we're going to talk about the mutation. All right, mutation. So, so from now on, we're going to spend probably like a couple of like lectures to talk about the genetic dissection of mutations. All right, so let's look at first. Let's see what is the mutational analysis. All right, so the what is the genetic dissection of mutations? So there are different categories of this kind of dissection. The first one is called the mutational analysis. So this one can be deemed as a passive study process because we just don't do anything to make change to the organism. We just observe. All right. So in this study process, the mutational analysis. So this is a process of analyzing the phenotypes of organisms known to be a mutant. Okay, for a particular wild type allele, we know this is a mutant. Then we will put this organism under the study. So this is a passive dissection, genetic dissection of the mutants. All right, there's another category, which is the mutational dissection. All right, this one actually is deemed as an active study. So we will intentionally disrupt some wild type alleles in the study to study on the effect of that mutation on the organism's phenotype. So we will intentionally do something to produce mutant from the wild type to see if this mutant can have some different phenotypes. All right, and also if we do not know the relationship between the mutant and the specific phenotype, so we just put the organism under the stress or condition with the mutagens, and we're going to observe. So what the new phenotype will come out from this kind of experiment. So this is the third type. All right. Okay. So we have studied this before. If we look at a single gene, for example, this is a single gene, and this is the wild type allele, the capital A. All right. If there's a mutation occurring here, so this will generate a mutant allele, right? So within the same gene, it is possible that a mutation could occur here or here. So in this case, we will have multiple alleles, but all these alleles with the mutation will be called the mutant alleles, different forms of the same gene. And we have also studied this case. So for example, the wild type flower is a purple color in the flower petal. All right, and this actually this wild type phenotype is determined by two genes, W1 gene and W2 genes. And if the biological pathway giving the wild type phenotype is like this, so suppose this is the pathway, and starting from a colorless precursor 1, and the first gene will produce a enzyme, the first enzyme 1, and this enzyme will transform this colorless precursor 1 into the precursor 2, which is also colorless, and then the wild type gene 2, which is W2, the wild type will produce a functional enzyme 2, and this one can transform the colorless precursor 2 into the blue pigment, and which will be deposited in the flower petal, and then we're going to see the wild type phenotype. Okay, so this is the biological pathway behind. All right. If this is a mutant, and if this mutation occurs in gene 1, and this mutant the same, the mutation occurring also in the first gene 1, W1 gene, but in a different location, right? But for the third mutant, so the mutation actually occurs in the gene 2. So different, right? Different allele, different mutant. Okay, now if we do the complementation test, we have learned this before. I hope you still remember what the complementation test is, and in what situation we're going to use this application tool to help us to dissect the genetic mutation, the genetics of the mutation. All right. So if we cross these two mutant, we still get the mutant. Why? Because for this two mutant, the mutation occurs in the same gene. So these two mutants cannot complement each other. So that's why if you cross these two mutants, you still get the mutant. And so if this is the result, immediately we know, okay, so the mutation occurring in the same gene for these two mutants, right? Okay, now if we cross these two, right? If we cross these two, mutant, we're gonna get a wild type. So why is that? Why is that? Because this is only because, all right, the mutations of these two mutants are occurring on two different genes, all right? So if we cross these two mutants, so for this gene, we got a heterozygous, right? For the second gene, we also get a heterozygous. Both are functioning, and the pathway is functional in this case, and then we're gonna see the wild type. So that's the complementation test. All right, now if we do the dihybrid cross for this individual, we're gonna expect to see this, right? However, owning this category, because these two genes are complementing each other, right? Complementing each other. So the relationship between these two genes are complementation to each other. Then these three classes will show the same phenotype, which is mutant, while which is the white color, and only this category will show the purple, right? This gives us this ratio, 9:7. Okay, question, let me see. So it is functional is because, okay, let me show you why this is functional. Okay. Oops. So for this mutant, it's not functional because this is a mutant. So this gene is blocked in this mutant, right? So if we look at here, all right, because the second gene is blocked, so the second step is blocked. So this is why we see the mutant phenotype, the white flower in both mutant. However, if we cross two, right? Okay, now the progeny from here is heterozygous in one gene and heterozygous in two genes. So in here we have W1 plus in the first gene. This is mutant. But for the second gene, we have W2 plus as well, right? This is W2 mutant. Okay, this is functional and this is functional. So in this individual, both enzyme is functional. And then the entire pathway is functional. And then you're gonna see the wild type phenotype. Yeah, I hope this is clear, right? Okay, now, so why mutation will be useful to us? So of course, to any organism, mutation actually plays a very important role in adaptation, speciation, and evolution, of course. So the mutation will produce the variability in any populations, right? Otherwise, yeah, every one of us will look identical. So that's very boring. But because of the mutation and plays this important and a fundamental role in adaptation and evolution, then it will make a beautiful diversity in any populations. This is to the nature. All right, also to science. So to our scientists, so the mutation are also very useful because mutations will serve as a very useful tool to help us to dissect different biological pathways or processes. For example, we can leverage mutations to study different biological pathways like DNA repair, DNA regulation, and protein synthesis, biological pathway and cyclotransduction process, and so on. So we have used this powerful tool to help us to understand the genetics in different biological processes. Okay, so as I mentioned, from now on, we're gonna study the gene mutations, which is the inheritable changes of the DNA sequences at 3 different levels. So first, we're gonna talk about the mutation at the molecular level. After that, we're going to talk about the DNA change, an inheritable DNA change involving the mobile DNA element, transposon, which means a small DNA element that can jump from here to there within the genome. And this jumping and the insertion or leaving from a specific genomic loci can produce mutations, right? And also, after that, we're going to talk about the chromosomal mutation. So the mutation involves the entire chromosome, either missing, duplication, or recombination, something like that. Okay, let's look at the mutation at the first level, the molecular level, point mutation in DNA. Okay, so this slide shows a lot of important, useful concepts. So please pay attention because in test or in the quiz, I may give you some questions to test on these concepts. Okay, let's see the first example, the wild type. There's no mutation at all, so you're gonna see the standard DNA sequence here, right? And each 3-letter so this is called the codon and will code a specific amino acid. All right, so this is the wild type, and the wild type will produce a protein with a specific sequence of the amino acids and to form a functional protein. So this is a functional protein, and this protein is functioning normally in an individual which produce the wild type phenotype. Okay, this is the wild type. Okay, so if there's a mutation here, so if the point mutation, so for example, if this letter, this SNPs, this nucleotide has been changed from A to C, as you can see here. So if there's a change, however, this change did not change anything in the codon. So still, even there's a letter change, right? So still, as you can see, the codons will code the identical sequence of the amino acid. So alter the codon specify the same amino acid. So this will not change the gene product at all. So if this mutation is like this, so we say this mutation is a synonymous mutation, because even there's a change, even there's a mutation, there's nothing changed in terms of the protein function. We can still, so this individual can still produce the same functional protein, wild type. Okay. All right. Now, if the change of the nucleotide will specify a different amino acid. However, if this amino acid has a very similar chemical feature with the original one, so even there's a change in the codon amino acid, right? But the resultant protein have the same shape feature of the wild type. So this will also give you the wild type. Then we say, okay, this is missense mutation. However, it is conservative missense mutation because the change of the amino acid will not change the feature of the resultant protein, wild type. Okay, so this is the conservative. Now compared to this one, so this is non-conservative. So the change of the nucleotide will specify a amino acid which has a distinct chemical feature when compared to the original protein amino acid here. So this will totally change the feature or shape of the protein, and this will lead to the mutant phenotype. Question, let's see. All right, so this is this is a very good question. So how can we differentiate conservative and non-conservative amino acids? So I will not test you on that because it's too difficult. But so in the test, probably I can ask you to give me the definition. So what does the conservative or the non-conservative missense mutation mean? So you need to understand the difference between them in definition, right? So first, the missense mutation means like a change in the point change mutation in the DNA sequence will specify a different amino acid. However, in the conservative missense mutation, even there's a different amino acid, it will not change the shape of the resultant protein. Still can function like the wild type protein. So that's the conservative. Means like even the change of the amino acid will not change the function of the final protein. All right, okay, you're welcome. So the non-conservative means like, okay, this is not the case. So if the change of the amino acid will totally change the feature of the resultant product, the protein, which will produce the wild type phenotype, and then that's the non-conservative, right? Okay, let me erase this. Oops. Okay, so the nonsense mutation means, okay, so if there's a single point mutation occurring here, and if this change in DNA sequence will introduce a stop codon, then totally the function of this gene will be destroyed. So this is the nonsense mutation. Okay, and this, of course, this will reduce the mutant phenotypes, right? Okay, so those concepts are very important. Yeah, everyone should understand the difference between the concepts. Okay, and there are some other concepts here, the transition or the transversion. Okay, so we all know like, so T and C belong to the pyrimidines, right? And A and G are the purines. If the mutation occurring between pyrimidine and the pyrimidine, then, or if the mutation occurs between the purine and purine, then we say this mutation is a transition. However, if the mutation occurs between the pyrimidine and the purine, then this kind of change mutation is called the transversion. Right? Okay, so this is for the mutation, and there are other forms of mutation here. So either the insertion or deletion. So the indel here means like, this is a combined word. It's the insertion and the deletion. Okay, so insertion means like if we have a single nucleotide insertion here, so this insertion will totally change the reading frame, right? The frame shift mutation. It can totally change the codon for the sequence of the amino acid. The same thing here. So if we remove the one letters, this kind of removal of a single nucleotide will change the reading frame. So the frame shift mutation will totally change the function of the resultant protein. Okay. Oops. All right. So the mutation can occur in the germinal mutation, in the germinal cells or the somatic cells. So the germinal mutation means like this mutation occurs within the gametes during the formation of the gametes, and so this mutation will be affecting the entire body. So since the mutation occurs started from the germinal cells, right? So then this mutation will be passed to every single cells in the whole body. However, some mutation may occur in the somatic cells, so this can only affect a proportion of the cells, not the whole body. And the mutation could be a forward mutation, which means like the mutation can result in a new mutant phenotype, or the mutation can be a reversion, which means there's a mutation, and then there's another mutation can reverse the first mutation, right? The reverse of the new mutation. So this mutation it will be called the reversion mutation, and the mutation can have different kinds of effect on the organism. Either the change in the either the morphological change, or the biochemical change, or the physiological change. And we have learned this before, is the mutation can cause the loss of function, or gain of function, or suppressor mutation. So I hope you still remember this concept. The suppressor mutation means we have two genes, right? So A gene and B gene. Okay, so the A gene mutation will cause some mutant phenotype. However, if B gene is also mutated, so this mutation could be a suppressor mutation, and the mutation in B gene may be able to reverse the mutation in the A gene. So I don't say like definitely the mutation of the B gene will reverse the mutation of the A gene. I say it could be like that. The mutation in B gene can reverse the mutation of the A gene. All right, we have learned this before. Okay, so okay, so for example, in this case, this is a wild type of the gene, and this wild type can produce a functional protein with a specific shape, because the protein usually is functioning with a specific shape in the cell. All right, and the pink area actually represents the most active parts for the protein. So any change in this pink region will be fatal. So it will severely affect the function of this protein. However, if any mutation occurring in some not that important region, so this mutation, for example, will not affect the function of the protein. So we say this is a silent mutation, for example. But if mutation occurring in this pink area, so this will cause the dysfunction of the protein. So and this mutation will call the non-mutation, right? So we have learned this before. Okay, so let's see. The loss of function means, okay, so the wild type allele can produce a functional protein, and this functional protein will result in the wild type phenotype. If there's mutation occurring in this gene, so this allele cannot produce any functional protein. So in this case, we say the loss of function, right? Okay, and if for example, if a mutant allele so still can produce some functional protein, but as you can see, the amount of the functional protein produced by the mutant allele is significantly less than the wild type. So in this case, we can say this mutation will cause the leaky loss of function of the wild type. So yeah, there are two different concepts here: the total loss of the function or the leaky loss of function. And the gain of function is very easy to understand, right? Okay, so this is the wild type. So for example, the wild type allele will produce the purple pigment, which will show the purple flower in the plants. All right. If there's a mutation, let's say the M, and this M will produce yellow pigment, then you're gonna see this yellow color in the petal, the flower petal, right? Right. So this means like this mutation will cause some will cause gain of function. This is easy to understand. All right, so I think I'm gonna stop here today. So any questions so far? In terms of mutation?
I had some questions about the previous slides.
Previous slides here?
On the lecture 9 and 10.
Oh, okay, sure. Let me stop sharing first. And okay, this one. Sure, which slide?
I believe it's slide 69. It's the one about the squash with the dominant epistasis.
Here?
Yeah.
Okay.
Yeah. So I just wanted some clarification. So capital W inhibits any color. So that would automatically make it white.
Okay.
But then is lowercase w a precursor to either capital Y yellow or capital Y green or lowercase y green?
I see. So you need some explanation in this pathway, right? Okay.
Yes.
Sure. Okay. So let's look at here. So here, so we are talking about two genes. So one is the W genes, the other one is the Y gene, right? So first, we need to understand the different functions of these two genes. So the W genes actually, so either the capital W or the lowercase, right? So the W means, okay, so this enzyme, so this is the second gene. Okay, let's look at here. So the W gene is working on the colorless precursor, right? So if it is a dominant or wild type, the capital W, so it cannot. So that's why I put a bar here. It cannot transform this white, the colorless precursor into the green. Okay, it cannot. Only if the W is a lowercase, it can. All right. So this is the biology behind. Okay, so that's why. Okay, because the capital W cannot transform this pathway, right? So you're gonna see these are all white because this step has been blocked due to the capital W. Does that make sense?
Yes.
Okay. All right. So let's look at here. So if the W gene is a lowercase w and W, then this step is okay, right? So the white, the colorless precursor will be transformed into green, all right? Okay, now, so if this is the case, then this Y gene will start to work on this case, all right? Okay, now we have capital Y and the lowercase y. If it is a capital Y, then this enzyme will continue transforming this green stuff into the yellow. Then you're gonna see this, so the yellow. Okay. However, if this Y gene is lowercase y, so it cannot do anything on this green stuff. So the green stuff will stay here. That's why you see.
Okay, that makes sense. So capital Y would make it go through the entire line.
Mm-hmm. That's right. Yeah. So this is a good question actually. As you can see, in this case, we have what's the ratio? Let me erase this. So this is the dominant epistasis, right? Oops. That's why the dominant of the first gene will totally override the second gene, the function of the second gene. So that's why you're going to end up with this ratio. So if we go back to this case, so that's the recessive epistasis. So which means like only the recessive of the first gene will override the function of the second gene. Does that make sense? So
yes.
that's why you're gonna have different ratios, 9 to 3 to 4.
Okay. I also had a question again about the dominant suppressor mutation with the purple and white flowers.
Let me see. This one, right? Oh, this one?
Yes, that slide. Okay. So I'm still a little confused on the P plus SU, the second line. Mm-hmm. So I understand how it can be white because the SU can be the suppressor mutation alone, and that mm-hmm. would make it white. Mm-hmm. But would it be purple if, like, is the P plus overriding every, like, the SU? Is that why it's purple in the 13:3 ratio, or?
Okay, so this depends on, so different ratio depends on different story. So actually here I gave you a story about this ratio, right? So let me give you another reasoning about this ratio. Okay, so let's say suppose the P plus will produce a protein. So the protein itself can determine the purple flower, right? So suppose the wild type P gene will produce some protein which can work by itself to produce the purple pigment, right? Then this will gives us the purple, right? Mm-hmm. Okay. All right. Another case is like, so if there's a mutant of the P, it can produce a protein that cannot work by itself. So it needs something to be bind such that it can produce purple. So if this is the situation, then this makes sense, right? So the P plus itself can produce purple. The P plus cell itself can produce purple, but this cannot. Only if, like, the suppressor the suppressor genes also be a mutant, then it can produce a perfect enzyme that can bind to this protein, then this can produce the purple. However, if this mutant P gene produce something like this, but if the suppressor gene is a wild type, it cannot produce an enzyme that can perfectly match this protein, then it will cause the mutant phenotype. Does that make sense?
Yeah, so for the 13:3 ratio, mm-hmm. Would you define the P plus in the second row as haplosufficient since it can become purple by itself?
So this is a good question, but I think it's very hard to use the insufficient or sufficient concept in this situation, because haplotype, haplosufficient or haploinsufficient, usually we talk about that concept when we talk about the interaction between different alleles for one gene. So here we talk about two genes. So maybe, it's not easy to use that concepts here.
So if you were to test us, would you just give us a ratio and then ask us how it might have gotten there?
Yes.
Okay, thank you.
Sure. Any other questions? Okay, so if you do not have questions, we're gonna stop here and see you next time. Thank you, everyone. Have a good day. Bye-bye.